All right. Since some of you in your internet mungkin not working that well, so I record. So let's say ada yang terkeluar termasuk, you can always go to the recording version. Okay. All right. So our class today is on genetic modification tools. All right. So basically, you learn the basic techniques of genetic engineering, which is cloning. All right. So cloning is a very powerful method. However, beside cloning, there's other tools that you need to know in order for you to modify gene basically. So today we're going to learn a few of the tools and the model organism that you need to use in order for you to make sure that whatever you are genetically modified, you can be shown in something. Because sometimes, whenever you want to improve genes or you want to improve some traits inside bacteria, human, plants, you need a model organism so that you can study how does the changes make to the organism. So we're going to learn a bit about the model organism that we use in genetic engineering and also what kind of genetic modification that we can make. All right. So basically, previously you know that mushroom that don't basically sebelum ni tak pernah dengar pun about mushroom that don't go brown, mist that kills insect, medicine tailor-made for the individual. So if you seen the statement, uh, have you ever wondered that whether or not those statement will be a fact or is it just a fiction? Macam whatever you saw in any science movie, let's say macam apa science movie ni macam apa tu cerita tu yang mutant tu X-Men. Alright. So we never know that there's someone that has that kind of superpower macam the Hulk, right? So those are the things seems to be very fictional, all right? But of course, since we know that the technology has been evolving, and we know that there are chances that we could get this kind of traits in future, all right? Do you agree with me, or am I alone? Agree tak when I say something like that? Kan? I mean, dulu we never thought that okay, give me one example. Anyone can type any example that you think dulu you never know that these are the things that could happen, but somehow due to current technology, we found a cure or something like that. Anyone with E. coli production? Ni mesti tajuk thesis FYP ni baru baca. Baru baca literature review. Okay, that makes sense. We never know that bacteria can produce a plastic. Yeah, before this, right? Other examples? Any more? Any more examples? Semua dah diam dah. In vitro animals, transgenic animal, yeah. What kind of transgenic? Marijuana can cure cancer, alright, because of the active compound, yeah. So I mean, before this, cancer is smoke weed every day. Smoke weed every day. All right. So before this, you never heard yeah, some golden rice. The salmon tu, apa nama that salmon? I tak ingat lah. The salmon that genetically modified, that bigger with a very big good quality flesh that people sell nowadays. Yeah, the super salmon. Thank you, Chayi. All right. And then there's a lot more that you never imagined before can happen. But nowadays we can see the evolvement, especially in medicine worlds. All right. I think the first thing that come in mind when people try to modify gene or do some genetic engineering towards the genetic is to cure disease that are lethal ataupun that harmful to humans. That's why people try to find cure for it, something like that. That's why gene therapy experiment done. Right. So those are the things that we could say that it could be a fact rather than a fiction. All right. Mungkin nowadays you couldn't see it now, but people are trying trying their best in their research to make the all this kind of statement to be a fact rather than the fiction. All right. So genetic engineering is that kind of tool. All right. We try to modify genes in a way that we could get benefit on it. All right. Of course, there's some ethic issue lah on it. So later we learn about some ethics issue. But now I won't touch about it. I won't touch about ethics yet. We just go with the flow on the tools first. Okay. So as an introduction, every living thing is made up of the same building blocks, or we call it cells. All right. Cells work together to make an organism function, grow and survive. And then scientists use genetic engineering to modify cells so that the characteristic of an individual organism may be enhanced or modified. All right. Such engineering can be applied to any organism, from a virus to a plant. So basically, these are all the things that I tried to explain in previous slide. So it's in the introduction part. All right, so what is actually genetic manipulation? Genetic engineering is a direct manipulation of the DNA within a gene to modify an organism characteristic in a certain way, meaning that you wanted to see in terms of phenotype. All right, genotypically modified so that you can see the expression. So you nak tengok benda tu, tak naklah modify internally that it doesn't give any effect in output. Alright, so you want to see the phenotype in certain way. Alright, genes are small section of the long DNA sequence that is ordered into chromosome in animal and plant cells. So gene ni what is gene? Just a recall, I know you know what gene are. It's actually a small section dalam your DNA. If let's say you have a long DNA, alright, so gene is only a small fraction. Alright, so you learn about lag operon kan before? Kan? Lag operon tahu? Operon? Tahu. Okay. So dalam lag operon dia lag Z, lag Y, lag A, lag O, berapa banyak lag. That is gene, meaning that multiple genes are responsible for one operon. So those genes play a significant role that we can manipulate. Alright. Okay. So adding characteristic to a living organism such as resistant to drought or disease can be achieved through modifying this gene. So meaning that if let's say you wanted a maize that can tolerate per draw very limited of water, so you try to implement gene for water resistancy inside those plants. So this is what scientists are doing actually for plant like paddy, corn, something like that, right? So that we can increase the performance of the plants, all right? So there's a lot of method actually for genetic modification. So for method for genetic modification have rapidly evolved over the last 100 years, from basic animal husbandry and inserting gene from one organism to another more advanced method of directly editing the genome. So currently the famous one is CRISPR. So by now you dah tahulah CRISPR tu apa. CRISPR ni you can cut wherever you want. Something like that lah. You can cut the gene wherever you want. Kalau previously kita need a selected location, we must make sure the gene duduk dekat tempat yang RE boleh cut, something like that. But now, you can cut it whenever you want. Alright, so how advanced is the technology right now, okay? Alright, so one of the method that you learned before is using the plasmid, alright, where you insert the gene of interest inside it and then you clone it, alright. So plasmid method, I tak elaborate more, you can read, or is basically the same like cloning method that I explained before, alright. This is plasmid method, alright. So this one is one of the example of the cloning processor. So you can just read this. I think I also include it in previous lecture during cloning. You guys can read it again, alright. So, and then another one is using vector method. Alright, so previously kita pakai plasmid, now a vector. So, vector not necessarily a plasmid. It's basically a vehicle where we can transfer a foreign genetic material from cell to another. So, vector are typically viruses that attach to a cell and insert its DNA into the host cell and then detach itself. So, in other words, beside plasmid, we can always use a vector method, which is from a virus. So, virus tu, kita akan insert extra gene inside it and kita akan infect it toward the host cell so that the desired organism that we wanted the gene to be inside get infected from the virus. All right. Then the genetic information will be transferred to the host cell. So this is a vector method usually applied to higher organism macam plants and also yeast or even cyanobacteria or microalgae, those kind of organism. Alright, so this is examples how how vector method used. Alright, using a viral plasmid to generate a virus, and then you have a virus, you have a viral plasmid, then you deliver to host or viral packaging so that the virus could carry the inserted gene that you like. Alright, and then dia akan infect others and then virus with modified genome. So the virus will be modified and then dia boleh infect other and insert it additional gene inside the host. Alright. So this one you boleh baca je lah. It's very straightforward. This one very straightforward method for gene transfer using vector virus vector. Alright. Okay, so this is one of the good paper. Alright, if let's say I ask example of a process for viral vectors for gene therapy, so this paper can be your answer. Alright, try to summarize it and try to answer in a simplified way how they do it. Alright, okay, so nanti kalau ada masa baca, kalau boleh baca. Alright, it's good for your own knowledge. Okay. And then a third one is biolistic method. So apa kita kata dengan biolistic? Bila biolistic ni, the name called for fire shooting. Alright, we use gun in order for you to insert the DNA. So what they do is uses of gene gun to fire DNA particles into a cell. DNA can adhere to extremely small particle of metal such as gold, silver, and tungsten due to its its polarity charges. Alright, you know that DNA has charges. Sometimes it can be more positive or more negative based on the amino acid composition inside it. Alright, so based on those charges that they have, we can use a biolistic method to transfer the DNA. Alright, the DNA coated metal particles are placed inside the gene gun. A slight vacuum is created between a gun and the tissue, and then DNA particles are shot into the targeted cell. This is typically used to modify plants, although the science evolved include animals as well. So usually biolistic method ni orang buat dekat tissue culture. Alright, orang buat in plants. Alright, so it easier method for plants. I mean kalau bacteria it be easier lah because we use plasmid. But for plants mungkin susah sikit nak do a cloning. So biolistic method become a common approach in plants. Alright, so this is basically the picture. Alah, tak cantik pula picture ni. Alright, dipecah. So basically you have the DNA, you load the DNA inside the tubing. So the tubing are rotated and then you letak tubing to inside this gun. Kalau you nampak rupa dia, I tahu tak cantik sangat, but you can Google the image by yourself as well. Then you load the cartridge inside the gun, then you deliver DNA into the target. So you tembak je dekat chloroplast. Alright, so this one nantilah kalau I jumpa I ganti yang lain. I don't know why dia pecah kat sini. Alright. Okay, so this is biolistic method. Alright, so boleh Google, ada YouTube juga. So kalau you perasan, this is how the DNA this is how the DNA ni, kejap webback keluar apa ni. Alright. Okay, so basically gene gun barrel. So you loaded the DNA here. So they are de-stopped by a screen, I think. Then plastic disc with DNA coated gold particle. So kat sini punya DNA coated. So bila gun tu tembak, then the particles will go, the DNA gold particle will be inserted into the plant cell. Alright. Alright, so these are the more better tapi pecah juga. I think waktu I nak ambil tu, gambar tu I kecil kot, resolution dia kecil kot. Nanti kita cari yang baru. Nanti I put a new one. Alright. Okay. So this is very straightforward. Gun method is very straightforward. Ada pun gambar. Okay. So this device inject cells with genetic information using heavy metal element bullets coated with plasmid cells. So nampak tak? Dia macam a normal gun. So very straightforward. Nampak macam Appendix punya pipette yang multi-channel tu kan. So it's as easy as that. Alright. So biasanya untuk plants very easy. You just blast macam tu je. See? Sebab you ambil the tissue from the leaf, you tembak dia, then you put on top of the a new agar plate for the tissue culture. straightforward. All right. So gene manipulation is okay. Alright, so gene manipulation is a technique that involves the manipulation of genetic materials. This is just the explanation of the tool lah. I usually memang tak suka baca lah kalau ada long paragraph. So you guys can read because I give you the lecture note. Unless kalau I tak bagi, I tolong baca lah. Alright, so this is some, okay. So when you have the method to insert those DNA, you need to prove whether or not your gene manipulation works or not, right? Right. So when you want to prove it, so you need to put it inside a organism in order for it to develop, in order for them to the gene to be expressed. So what kind of organism that we will use? All right. So there's a few model organism that are well studied. I rasa banyak you dah tahulah basically. This is quite common. The first one we call Saccharomyces cerevisiae ataupun baker yeast, the one yang mak you buat jemput-jemput tu. I don't know whether your mum use jemput-jemput masak jemput-jemput ke tak, tapi I use that one. Or if your house have Thermomix, the one that your mother do pot roti, it's not just Thermomix lah. Kalau you ada bread machine ke kan, kan you use the yeast, kan ada satu bau from the baker yeast tu kan. So these are the most common fungal that was studied. Alright, so meaning that we understand most of the mechanism inside the yeast. That's why it's become a model organism. So kenapa yeast? Why yeast in the first place? Alright, why? Because yeast is unicellular. Alright, it can when it's unicellular, you can represent a higher organism punya examples. Alright, if let's say you want to study it, in unicellular organism, so yeast is one of best option because fast, rapid growth and easy to culture as well. And then one of the smallest eukaryote genome, so meaning easier for you to see. Let's see kalau gene tu almost similar that you can be insert it dalam yeast, it is easier for you to see how the changes the gene can make, right? Exists in both haploid and diploid. So this is one of special characteristic of the yeast. And then many cellular processes are conserved to humans. And then at the same time, the cellular processes are lebih kurang human punya cellular processes. Alright, so the carbohydrate metabolism is almost similar. So you can see the changes almost happen in human as well. That's why they use yeast. Alright, then this one is the worms ataupun kita panggil Caenorhabditis elegans. Alright, so this one also quite famous. Tapeworm atau worms lah. Alright, so why worms? Sebab dia simple. So sometimes you want to study in animal-like structure, right? So when you want those kind of activities, when you want to see the expression, you can simply take worms. Why? Sebab dia simple and easy to grow. It's very transparent. You can see what's happening inside the cell, inside the gut. Alright, because Caenorhabditis ni dia punya morphology is transparent. Alright, and of course it's cheap and easy to grow, and you can freeze it to regrow it later. Alright, and then when you study relationship with human disease, people understand that 75% of human disease gene have potentials Caenorhabditis elegans homologue. So meaning that when we have a homologue, it's similar to human. Alright, meaning that even though it's a worm's gene, however, the gene sequences is homologue to human, meaning it gives the same annotation. It gives the same function with human gene. Alright, so in this case, that's why we use worms to study human disease as well. And you can see that 40 to 50% have a C. elegans autolog. So autolog is same ancestor. Alright. So there's a few terms that you will learn later. I don't know whether we learn kat sini ke tak. Tapi we have homolog, paralogs and autologs. Alright. So this log indicate how the relationship of the gene between the ancestor or the genome, between genome A and genome B. Alright, so those terms is your task to find. What are homologs? What are autologs? But I do explain it just now. Alright, autolog and paralog, so that to make things clear between you guys. Alright, so make additional notes on homologs, autologs and parallax. Okay, so for C. elegans, it has 40 to 50% autolog, meaning that it carry the same ancestor gene as human. It's similar to human, 40 to 50% genes. Alright, genetically fixed number of cells, so dia memang the number of cells are fixed, so it's hermaphrodite, ada 959 somatic cell. If it's true male, it have 1031 somatic cell. So meaning that the cell number is consistent, easier for scientists to manipulate or to study the changes of the cells later. Alright. That is worms. And then this one, the famous one, which is Drosophila melanogaster. Okay, so for Drosophila melanogaster, susah nak sebut nama mereka ni. Drosophila melanogaster, for this one is actually a major animal experimental model. So kenapa Drosophila ni kita suka? Why we study using Drosophila? Kejap ya. Okay. So Drosophila ni, it has a special characteristic. Short breeding time. Betul-betul, Eugene. Fewer chromosome, yes. Ya. Ini pernah buat intern kat sini dulu. Dengan vector parasite. Alright, so basically, fruit fly ni, kenapa kita know? Year 1 project. Okay, good that you still remember. Alright, so the lecturer play a very important things there. You still remember the fact. So, tahniah for that lecturer. Alright. Who teach you about Drosophila? Dr. Hadura? Dr. Hafiz? Saja tanya. Dr. Dalina. Okay, then good to know. Dr. Dalina teach you well. That you still Dr. Mano. Mano is who? I don't know lah. Is he from our school? Dr. Mano? Oh, Prof. Nazalan Najmuddin. Alright. Good to know. Oh, you guys still sempat dengan Prof. Nazlan, then good. Alright. So basically, why fireflies? Because fruit flies share nearly 60% of human genes, meaning that sama. Alright. When it's similar, it's easier for you to study because you have the same gene. Alright. Fruit fly and human use the same similar gene to develop into adult. So for fruit fly, since the development genes is similar, it's very good for a researcher to know, to understand. If let's say they manipulate a certain gene that evolve in the growing, you can see the changes. If let's say you manipulate that, you know what happened next. All right. So we can predict in human as well. All right. And then 75% genes known to cause disease in human have autolog in Drosophila. Autolog means it carry the same gene. Alright. Kalau homolog, different sequence, same function. Kalau autologs ni, maksudnya sama. It's the same. Alright. Respond to alcohol. Okay, the good thing about Drosophila, ni key dia. It respond to alcohol, nicotine, crack and cocaine. Meaning that when you minister drug to them, they respond to it. Alright. I take a 5-minute break. Kat rumah I ada azan. Wait lah. Let pause for a bit. Nanti kita sambung. Alright, okay. So last point for Drosophila is quite cheap and quick, alright, because it's short lifespan. So basically, kalau you guys perasan, all the model organism dia ada very short lifespan, so that it be easier for researcher to study the before and after effect. Kalau kita guna animal yang lifespan dia panjang, contohnya macam let's say you nak study in cats. Cats kan dalam 15 years kan, contoh. Alright, so takkan nak tunggu sampai cat mati 15 tahun baru nak tahu effect the gene. Alright, so those are the things yang researcher take into account lah whenever they want to study the effect of the gene, the modification that they have done. Alright. Right. So this one is okay. So this is the famous one. It's more higher organism, and at the same time it possess a similar gene with us. Alright. So this one kita panggil dia siapa? Musculus ataupun mouse. Alright. So this is example of how mouse cell tissue culture look like. Alright. So why mouse? Okay, dia sangat famous lah mouse ni. If you remember, I think waktu early my slide dulu I ada bagi CV dia. Semua guna dia. Alright, so it's very very famous model organism. Why mouse? Sebab dia close to humans. Alright, very close to humans. And then it's a mammal. Alright, so we can observe the changes easily. And then you have a complex systems like human. Okay, contoh dia ada endocrine system, immune system, nervous system, rather than macam yeast, dia tak, it doesn't possess those kind of system that make Drosophila pun tak ada. Alright, so it's hard for us to see the changes in, if let's say you want to see inside nervous system. So that's why mouse is the best model organism that we could have that can mimic human. Alright, and then protein coding region are 85% identical, meaning that it's very similar to human, 85% sama. Alright, and then the gene conservation are very high. Some protein are up to 99% identical. So in this case, 99 identical to human. Alright, the percentage is very high. Meaning that if you manipulate dekat mouse macam tu, then that's why, and the changes in human you can see almost similar. There's a very low there's very high gene conservation there. Okay, the last I think this is the last one. So the last major our model organism that that people oftenly use are Arabidopsis thaliana. This is plants. Alright, for Arabidopsis thaliana, it's always become a model organism for tissue culture ataupun genetic modification in plant. Alright, why Arabidopsis? Because it's small and easy to grow and it have short life cycle. Dia ada 6 weeks life cycle je. Alright, so it has a massive seed production up to 10,000 per plant. So if you want to study the seedling, it'll be easier because it have a huge seeds production. And then it's the genome is very simple, and yet it's more it's similar to more complex plants. All right, they're smaller sizes, but it's similar to complex plants. So to make it researcher can understand well how plant evolve. All right, easily transform with Agrobacterium tumefaciens, and then you have a vector that can carry gene of interest for you. In this case, we can use Agrobacterium tumefaciens to insert the gene of interest, and of course the ballistic method also can be used. All right, so that is Arabidopsis. So there's a few. All right, so if you want to watch more details on the organism, I give you this link, so have a look later. Okay, alright, okay lagi ke semua? Okay kan, sikit lagi, tak banyak dah. Ada forward and reverse genetic. In this class we focus on forward, tak silap forward dulu. Next class we focus on reverse. Alright, okay, so you dah tahu tools, and then you dah tahu model organism that you can use, and then you, of course, when you want to do changes, you need to know what kind of changes that you want to do for your gene. Alright, so there are two types of genetics that we're going to learn. Alright, one we call a forward genetic, another one is reverse genetic. So apa tu forward and what is reverse? Alright, so forward is actually you discover gene that contribute to the phenotype, meaning you do the changes in gene rather than the phenotype. And then you do daripada forward, daripada gene, then you observe the phenotype changes. And then kalau reverse genetic, you tengok the phenotype dulu, baru you find the gene untuk you modify. You tengok phenotypic tu macam mana, then you think, okay, in this phenotypic biological processes, I need to control the certain thing, then you do a modification. So it terbalik lah, maksudnya you reverse it. Alright, so kalau forward genetic is commonly kita panggil a term for it is mutagenesis. Reverse genetic, the one that you engineer ataupun guna RNAi. Why? Because macam cancer cell, you already know the location of the cancer cell. So you want to reverse it, you want to cut the sequence because you already know what the effect of it. Macam mutagenesis, if let's say site-directed mutagenesis ataupun when a normal mutation happens, so those are all forward genetics, meaning that the one that you cannot control, right? If you control, meaning that you create the condition and you expect the performance. You expect what phenotypic that you can get. Alright, that is a control mutation lah. However, more kalau forward ni more kepada general or natural selection. Alright, let's see what forward genetics are. Right, it's actually a traditional approach to study the gene function that begin with a phenotype. Alright, maksudnya you tengok the mutant organism and you want to fix it and proceed to a gene that encodes the phenotype. Meaning, you tengok, let's say, macam in the case of transposon, alright, you see, people see that there's some corn that are black in color or white in color. That one is not the one that we purposely do mutation, right? So in that case, what Barbara McClintock do, he bro dah kesian Barbara McClintock. She thinks and see what are the difference between the gene that make the color changes in the corn. Alright, so that is one type of forward genetic. Meaning you see the changes, then you study the genetic modification that happen. So it depends upon identification and isolation of random mutation that affect the phenotype of the interest. You tak kawal benda tu. It's naturally happen. Alright, so that is forward genetics. Alright. Kalau reverse genetic is basically a molecular approach that begin with a genotype and then proceed to the phenotype by altering the sequence or by inhibiting its expression. Meaning that you nak elakkan benda tu terjadi. That's why macam cancer, you know the sequences, you tahu the outcome, then you want to cut the gene that is responsible for the cancer, and then you inhibiting the expression of the gene. So that is reverse genetic. Alright, it's possible due to the advancement in the molecular genetics. So the one yang modification, genetic analysis, genetic editing semua tu more ke reverse genetic. Alright, so kita go to forward genetic. So apa yang menyebabkan forward genetic? So this one you dah pernah belajar. Alright, cuma tak tahulah dia duduk kat bawah forward genetic. So basically a normal mutagenesis that can be caused by gamma irradiation that can induce chromosomal break, UV radiation, alkylating agent, transposon, retrovirus. So these are all the agents that can cause a mutagenesis inside a DNA. Alright, and then powerful. So this is an example of powerful mutagen lah. Alright, so why ninja turtles can talk? Anyone saw the video? Tengok ke ceritanya? Ke tak ingat? Ni zaman saya je. You all tak tengok Ninja Turtles. Macam mana lagu Ninja Turtles? Tak ingat lah. I watch it. Then good to know that I'm not alone in this journey. Yeah, TM. That is short name for Ninja Turtles, Teenage Mutant Ninja Turtles. But do you remember how this apa nama dia, Raphael, Michelangelo become a turtle with these properties? Yeah, there's some explosion, right? With chemical, with some mutagen lah kita panggil. So some chemical cause a very powerful mutation. Alright. Eugene, I don't need you to explain about who directed Ninja Turtle to me. I'm more focusing on the mutation that caused Ninja Turtle. Alright. Okay, I get it, Eugene. You wash. Thank you for telling me that we are in the same time frame of age, so that I don't think that I'm too young for you guys. All right. Okay. So basically, it's because of a strong mutation that caused them to become like that. All right. That is some of the examples of powerful mutation that can cause turtle to become like human. Okay. They can speak and whatever. All right. So some other example of normal mutation is gamma eration that induce chromosomal break. So tadi dah dengar tak orang kalau you pergi X-ray, okay, because there are radiation there that can break your DNA. So that's why kalau you mengandung, you cannot go X-ray because it can do some DNA mutation to your body. Alright. Of course, there are level of exposure lah, but usually we don't want to risk, alright, because of the radiation. Okay, so this is another example of UV radiation bila there's ultraviolet light. So it causes a timer dimer that give this kind of mutation. Alright, meaning that a mixed match mutation happen. Okay, when your DNA want to repair, it's additionally it's additionally encode A to the mutation region that cause a mutation. Alright, this is UV radiation and this is alkylating agent. So when you have EMS, nitrosoguanide, mustard gas, these all are alkylating agent that can cause mutation to DNA. Alright, so these are the example. This one, the example that I told you just now, transposon, or we call it a jumping gene, where segment of DNA can move from one region to one DNA to another region, contain insertion sequence for cutting and reselling DNA. Kita bagi transposis gene, complex transposon carry other genes. So that's why there's a mix and match gene expression in corn due to the transposon ability. Alright. And then retrovirus because dia carry the RNA, okay? It reverse transcribe RNA genome inside the host cell. The new DNA genome is integrated into the host cell. So virus usually dia bawa a lot of extra DNA with it, alright? Okay, so this is retrovirus, this is normal virus lah. Just for fun. Alright, so this is an example of the processes for retrovirus. Have a look on the method. Ada method dekat dua paper ni, so you can see. So basically a normal infection lah. Sebenarnya I tak payah explain. It's very straightforward sebenarnya when retrovirus approach the cell, integrate the RNA, RNA akan bind dalam DNA, so DNA akan amplify, so you have integrated viral DNA inside your original chromosome. Alright. That's it. Any question? Kalau ngajar online kan dia jadi cepat tau. Sebab mungkin tak nampak you all kan. I tak tunggu you all jawab soalan. Okay je. Alright. I rasa I'm not too fast. Kalau I'm too fast, I'm sorry lah. Dengar balik lah recording version lah. Sorry lah. I have a very upset stomach today. Okay. So anything else guys? Ada soalan? Kalau tak ada, don't worry. Today attendance are recorded in Webex. So tak payah scan apa-apa. Tak ada soalan ke? Any question? For Khamis, I let you know whether I'm okay or not. Kalau tak okay, kita buat online lagi lah. I'm sorry for that. You guys adalah masa nak rindu I sikit. Okay, I nak explain a bit about lab report. Okay, for lab report, I expect a full lab report. Alright. So maksud full lab report, ada introduction, ada methodology, ada result, ada discussion, ada conclusion and ada references. Alright, lab report is in group. Nanti I create the link for you guys to submit your lab report dekat eLearn. So the due is 2 weeks from yesterday. So the coming lab is your due date lah. Maksudnya Monday the coming lab tu your due date. So I'm going to create the link. So you guys, one person upload untuk representative for one group lah. Okay, let's say ada group member tak bekerja sama, tak nak buat kerja. You can remove their name and put a remarks in the lab report. If that's the case, okay. Tak nak lepas ni doktor sebenarnya dia tak bekerja, doktor kami dia buat. Saya cover tempat dia. Tak nak macam tu, okay. So if let's say that thing happen, tell me or remarks in your lab report, alright. Either way lah, I read your lab report and see. Result Pak Doktor report. the report, what do we expect? Okay, result part, I'm going to run your DNA hari ni atau esok. I'm going to give paling lambat this Friday the DNA result. Alright. So hari ni Mahir dah run group 1 to 9 punya DNA. Another one maybe lusa kot Mahir akan run kan. I give you the result of group of your DNA. Okay. So don't worry about it. Nanti I bagi result. I akan bagi gambar band lah. So you explain from that. Okay. And then cuma paling lambat Friday lah. Alright. You can go for introduction first. Okay. Result discussion, your own word. Question is another part. Okay. Jawab question dekat part lain. Okay. And then apa lagi? You all nak apa lagi? Any more question? Tak ada dah rasa. Introduction tu tak payah panjang-panjang lah. Introduction tu half page pun cukup. Sebab kan introduction ni selalu masalah. Selalu ada yang buat sampai 1-2 page. Ada yang buat sampai 2 page. Dah kan susah saya nak marking macam tu. Sebab markahnya let's say kalau total 10. Introduction tu dapat 1 markah je macam tu. Saya memang akan fokus on result discussion dengan question more. Alright? Kalau pemarkahannya. So tak payahlah too hard on introduction. Methodology pun mungkin 1-2 marks je. Katakan over 10. Usually I prefer you buat flowchart je. I don't read each word. So kalau you buat flowchart, it be easier for me to see. Alright? Okay. Boleh terus copy paste doktor punya ke? Oh, the method ke? Okay. Kalau you nak copy paste I punya, you need to convert it into past tense. For method. Alright? Tak ada isu pun. Apa tu? Listen. Tanda soal. Intro tanda soal. Kenapa dengan intro? Listen. Copy paste intro. Tak boleh, tak boleh. My intro, okay. So what you explain in your introduction, a brief description about DNA extraction and also the method that you will use that day. Ya, ada orang nak cakap ke? Ya, Aida? Aida tertekan ke Aida nak cakap? Oh, tertekan. Oh, malu. Okay, tak apa. Kalau nak cakap boleh tau. Apa? So basically macam tu lah guys. Introduction explain lah sikit your own word or your own findings. Alright? Anything else? Kalau tak ada, I nak stop. The result just now I bagi tahu, I akan bagi you all dekat e-learn ataupun your group, the gambar. Paling lambat hari Jumaat. Tu paling lambat. Spacing. Allah, you all spacing pun tanya I ke? Buat yang macam sebelum ni lah you buat. Macam tak pernah ada lab work dengan I sebelum ni lah. 4th year kan? I tak payah tulis lah format tu. Ikut you je asal I boleh baca. Kalau you buat I tak boleh baca, tu lain. Alright? Anything else? Tak ada I tengok. Tak ada. Thank you Alright. Okay, thank you everyone. I update you on Khamis punya kelas. I try to update paling lambat malam esok lah. I'll see my condition how. Okay? Nampak je mulut macam okay kan? Tapi tak. Ni semua penipuan. Right, okay. Bye everyone. I see you next time. I'm not sure when, but Khamis surely. Cuma tak tahu online atau physical. Bye. Assalamualaikum. And have a great day. Bye. Welcome. I kena baca sebab nanti kalau I end awal-awal kan you all macam sampai tidur tak baca. Saya thank you. Thank you for the wish. Get well soon. Thank you. Okay. Saya dah baca Alright. Okay. Bye everyone. Assalamualaikum.